Ada yang terkeluar sebab saya recording ni, suruh dia masuk balik. Okay? Sebab dia macam tu. Bila saya start record, ada yang akan terkeluar. So tadi apa, Razin terkeluar, so dia masuk balik. Alright. Okay, so for today's lecture, so we are going to look at, first of all, is the last lecture we stop here, which is Rna splicing. And then for today's lecture, we are going to look at structure, function and structure of the RNA. Just to remind, kalau saya terkeluar, tolong call ya. Saya tak tahu kalau saya terkeluar. Alright. Message saya takut tak perasan. So please call if I left the meeting. Okay, so structure and function of tRNA. So basically, as you know, what is tRNA? tRNA is transfer RNA. Okay, so the function of transfer RNA or tRNA is to bring specific amino acids, okay, to the ribosomes, okay, that reads the codon, okay, the ribosome will read the codon of mRNA and allows specific tRNA to bring a specific amino acid according to the codon on mRNA. Okay, so basically all the RNAs, mRNA, tRNA, rRNA are all produced from the process of transcription according to the information in the DNA. In the nucleus. Okay, so they are transcribed from DNA templates in the nucleus. So after they have they are produced in the nucleus, so they will leave the nucleus through the nuclear pore and enters into the cytoplasm. So in the cytoplasm, it will involve in the process of translation. So for tRNA, it is a single DNA strand. So RNA, they are single-stranded molecule. Kalau DNA double strand, okay. But for RNA, they are single strand. So the length is about 18 nucleotide longs, 80 nucleotides long. So kalau nampak kat sini, this is the structure of tRNA. Nampak dia macam double stranded kat sini kan. So they are actually one single tRNA molecule that fold into a clover leaf shape. Okay, a clover leaf shape. Kalau dalam exam kan, dia tanya describe the structure of the RNA, do not just write the shape is clover, okay, clover leaf shape, okay. So this is the structure of the RNA. It falls into this form, okay, a clover leaf shape. So function is to transfer amino acid to the ribosome. So it has two ends, okay. So this end is where the amino acid will attach to, and this is the anticodon, okay. So the anticodon is a triplet, okay, is a triplet of nucleotides that is unique to every tRNA. So it means that each tRNA will have different anticodons, okay, because it has to match on the codon of mRNA to bring specific amino acids, okay. So each tRNA molecule can be used repeatedly. So after one tRNA brings specific amino acid to the ribosome, it can bring another same amino acid to the ribosome. Kalau dia baca the same, kalau ribosome reads the same codon of the mRNA, okay. So these are the bonds, okay, between the bases, okay. So kalau guanine and cytosine, you have 3 hydrogen bonds. Uracil and adenine, you have 3 hydrogen bonds. Okay, so an amino acid attachment site allows each tRNA to carry a specific amino acid to the ribosome. So anticodons are written in 3' to 5'. Okay, it is written in 3' to 5'. 5'. Sebab kalau awak tengok kat sini kan. Sekejap. Okay, so let's say this is ribosome. Okay, and in between the ribosome you have the mRNA. Okay, so this is the mRNA. So the ribosome will have 3 sites. It will have 3 sites. Is the A site, the P site, and also the E site. The A site is where the tRNA will enter. Okay, tRNA will enter and brings specific amino acids. Okay, so it brings specific amino acid according to the codon that enters mRNA codon that enters into the A site. Okay, so kalau for mRNA, okay, so it is read in the direction of 5 to 3'. Okay, so the anticodon mestilah complement, faham tak? Okay, so the anticodon, the direction is 3' here and 5' here. Okay, so you have 3 nucleotides. Okay, see 3 nucleotides here. So these are the anticodons. So the anticodons, the punya direction there is 3' and 5' here because it has to complement with mRNA that start with 3. That starts with 5 and ends with the 3 here. So nak baca codon pada A site ni mesti daripada 5 ke 3. Okay, so nanti bila ribosome baca mRNA tu, mRNA ni akan move. Okay, dia akan ditarik ke arah sini. Dia akan ditarik ke arah sini. So nanti mRNA tu akan masuk and masuk. Gerak masuk ke dalam A site, gerak masuk dalam A site. So example anticodon that you can have, for example 3' AAG. Okay, 3' AAG, so the mRNA codon will read 5' UUC 3'. Should be a 3' here. So kalau tak ada dalam otak awak, you just add lah. Right? Okay, so here is just to explain to you how the tRNA, okay, is attached to the amino acid, okay, using the enzyme here, which is amino acid tRNA synthetase. Okay, so amino acid tRNA synthetase catalyzes the attachment of amino acid to their tRNA. So first of all, the amino acid and the appropriate tRNA enters into the active site of this enzyme. Okay, which is amino acid tRNA synthetase. Okay, so amino acid and tRNA enters into the active site of the synthetase. So in this case, the amino acid is tyrosine. Okay, so the enzyme is also specific for the tRNA and also amino acid. So in this case, because it brings in tyrosine, so tyrosine, so the enzyme is called as tyrosyl tRNA synthetase. So kalau macam maksudnya dia nak attach different amino acid to different tRNA, so it will use a different amino acid tRNA synthetase according to the type of amino acid and also the type of tRNA. Okay, so in order to attach the amino acid to the RNA, it requires energy. So the energy comes from ATP, okay, adenosine triphosphate. So using ATP, the synthetase catalyzes the covalent bonding of the amino acid to its specific tRNA. So the amino acid is attached to the 3' end of the tRNA. So kalau kita tengok balik, so okay, so the amino acid site, okay, attachment site, it is the 3' end here. Okay, is at the 3' end. So sebab tu lah sini pun 3, sini pun 5. Okay, sebab hujung ni 5. Alright. Okay, so the tRNA charge with its amino acid is released by the enzyme, which is the synthetase. Okay, so once covalent bond has formed between the amino acid and also the tRNA, so you have this molecule which is amino acid tRNA is released from the enzyme. Okay, so bila dah attach ke amino acid pada tRNA, kita namakan dia sebagai amino acid tRNA, this molecule. Okay, the product is amino acid tRNA will be released from the active site of synthetase. Okay, so this is the structure of ribosome. So the structure of ribosome, as you know, it has the small subunit, you have the large subunit, and at the small subunit, you have the mRNA binding site. So it means that once the mRNA enters, okay, it will enter here kat sini dulu. Okay, it will attach to the mRNA binding site. So for the large ribosomal subunit here, it has three sites: the A site, the P site, and also the E site. Okay, A is amino acid tRNA site. So this is where the tRNA that is attached to the amino acid will enter. Okay, kat sini yang akan basically the ribosome will read the codon of the mRNA. Okay, so it holds the tRNA carrying amino acid to be added to the chain. So for the P site, okay, is peptidyl tRNA site. So this holds the tRNA that carries the growing polypeptide chain. Okay. So for the E site is where the tRNA leaves, exit the ribosome. Okay, so these are the E site that you have to remember lah. So, all the three sites, especially the three sites at the large subunit of ribosome. Okay. Okay, so building a polypeptide is through the process of translation. So translation is the process that synthesizes polypeptide chain. Okay, so it has three stages, which is initiation, elongation, and also termination. So this is the, as you can see, roughly, okay, a diagram to show how translation occurs. So the large ribosomal subunit, the small ribosomal subunit. So this is the mRNA binding site. So the mRNA will be in between the large and the small subunit. Okay, so the A site, as you can see, it holds the tRNA that binds to the amino acid to be added to the growing polypeptide chain. Okay, so this is the P site, peptidyl tRNA site. So this one, it holds tRNA with the growing polypeptide chain. Okay, so bila dalam proses translation tu, dia sebenarnya, okay, so this is the growing polypeptide chain. So this polypeptide chain, okay, so it breaks the bond between this tRNA and also the polypeptide chain and transfer to the amino acid here. Okay, transfer here. So this one, now this tRNA in the P site is now free. Okay, so what happened is that this tRNA will enter into the E site and then exit keluar. Okay, so now you have the tRNA with the growing polypeptide. This one enter into the P site. Okay, now you have the vacant A site that reads the next codon here. Okay, so maksudnya dia akan baca tiga bases and then another codon and then another codon here. So dia akan bergerak ke arah sini lah. Okay, so the mRNA codon are read, okay, from 5 to 3 prime direction. Okay, and anticodon dia punya direction 3, 5 kena complement dengan mRNA codon. Okay, so the detailed process for each of the stages. So the first one is the initiation. So initiation, the process start with a small ribosomal subunit dulu. Okay, so small ribosomal subunit binds to mRNA and a special initiator tRNA. Okay, a special initiator tRNA dia fixed. Okay, dia mesti dia mesti baca codon pada mRNA that reads AUG. Okay, so the anticodon will read UAC. Okay, UAC here. Okay, So anticodon of initiator tRNA, which is UAC, base pairs with the start codon, which is AUG on the mRNA. So AUG is the start codon, okay. UAC is the anticodon that is complement with this start codon. So the start codon AUG will code, okay, for the amino acid methionine, okay. So methionine. So AUG, which is the start codon, will code for the methionine amino acid MET. So this methionine amino acid will be bought, okay, by the initiator tRNA, okay. So this is the initiator that brings methionine amino acid according to the start codon here of mRNA. Okay, so then after the initiator binds, initiator tRNA binds to the start codon, then large ribosomal subunit will enter. Okay, so the initiator tRNA will sit at the P site. Okay, the initiator tRNA will sit at the P site dulu, bukan dekat A site. Alright. So then a large ribosomal subunit is attached to produce a translation initiation complex. So this is translation initiation complex. You have the small ribosomal subunit, you have the large ribosomal subunit, you have the ribosome, you have the tRNA, initiator tRNA that brings methionine. So these are the translation initiation complex. So the initiator tRNA must sit in the P site. Okay, so now you have a vacant A site. A site now is kosong, okay, vacant. So it will read the next codon here. Okay, the next codon here. So that the next amino acid tRNA can enters into the A site. So daripada point ni sekarang ni, the next amino acid, the next tRNA that brings the next amino acid will always enter at the A site. Okay. Cuma yang bezanya untuk initiator tRNA akan duduk dulu dekat P site. Okay, so after you have formed the translation complex tu tadi, okay, translation initiation complex. So next, you will proceed into the next stage, which is elongation. So during the elongation stage, amino acids are added one by one to the preceding amino acid at the C terminus of the growing chain. Okay, so consisting of 3 cycle. Ingat lagi tak ni? C terminus ni, ada N terminus, ada C terminus of this amino acid molecule. You remember the chapter 1? Okay, last time macromolecule. So we have looked at the structure of monomers for carbohydrates, the structure of monomers for protein, okay, the structure for nucleotides of DNA, and also fats, can. So understanding of your knowledge from last semester, you must apply during this semester. So amino acid structure dia mesti ada C terminus M dengan N terminus. Mana ni? Okay. And this is amino acid kan? H, C, double bond, OH. Okay, so this is amino acid. Ingat lagi kan? Okay, so it says here during the elongation stage, amino acids are added one by one to the preceding amino acid at the C terminus here. So whenever you want to add a new amino acid to the growing polypeptide chain, akan berlakulah proses of polymerization, hydrolysis kan. So dia akan remove OH of this amino acid and the next amino acid. R group kat mana? R group kat sini. Lupa saya kat situ. Okay, R group. Okay, so dia akan remove lah. So this is, yang ni dia attach kepada the growing polypeptide chain yang yang attached to the tRNA tu kan. Okay, so this is the last amino acid here. Ini. So you always add at the C terminus over here. Okay, at the C terminus. So the process is hydrolysis that involve the removal of water molecule. So ingat balik yang belajar semester lepas. So this is the new amino acid that is attached to the tRNA that sits in the A site. Okay. So during the elongation stage, amino acids are added one by one to the preceding amino acid at the C terminus of the growing chain. Mesti dekat C terminus. Okay, C terminus. So the elongation stage consists of three step cycle. Okay, 3 steps cycle. Okay, so the first step is codon recognition. Okay, codon recognition. Okay, codon recognition, and then you have peptide bond formation, and then is the translocation. Okay, so kita tengok yang first dulu, which is codon recognition. So codon recognition, the mRNA codon in the A site forms hydrogen bonds, okay, with the anticodon of an incoming amino acid tRNA. So this tRNA enters into the A site. So the tRNA here enters into the A site. Okay, because the A site is vacant. So next is the peptide bond formation. So for peptide bond formation, it requires the enzyme peptide transferase. Okay, so peptide transferase catalyzes the formation of a peptide bond between the amino acid of the polypeptide at the P site and the new amino acid in the A site. So this is peptide bond formation. Okay, the last bond, okay, here between the growing polypeptide chain and the tRNA here, it will be broken. Okay, and then a new bond will be formed between the last amino acid here and the new amino acid that sits in the A site. Okay, so the polypeptide separate from the tRNA in the P site and binds to the amino acid of the tRNA in the A site. So that is peptide bond formation. Okay, so now here the tRNA that initially holds the growing polypeptide is now free, and then it will enter into the E site. Okay. Okay, so now is the translocation lah. Translocation proses yang mana tRNA berpindah site. Okay, dia berpindah site. Translocation, translocate. Alright, so the tRNA with the attached polypeptide in the A site is translocated to the P site, making the taking the mRNA along with it. Okay, so here you have the growing polypeptide chain that sits in the A site. So dia kena bergerak masuk ke dalam P site. Yang ini yang kosong kena bergerak masuk ke dalam E site. Okay, E keluar kan, so that is translocation. So this brings the next codon to be translated into the A site. So again, the process will repeat lah, because now the A site is vacant, that reads the next codon. Okay, so the previous tRNA that was in the P site is moved to the E site and released from the ribosome. So translation proceeds along the mRNA in the 5' to 3' direction. Okay, so nampak ingat selalu kena ingat this is 5' and 3'. Okay, so dia akan bergerak. mRNA ni akan bergerak ke arah sini untuk dia dibaca dekat A site. Okay. Okay, so the last one is termination. So termination, it has these steps. Okay, so elongation continues until a stop codon, okay, in the mRNA reaches the A site. So when the A site has the stop codon, either of these three stop codon, which is UAA, UAG, and UGA, kan? So bila either of these three stop codon enters into the A site, so that is the signal for termination. Okay, so it do not code for it does not code for any amino acid, and it acts as a signal, okay, to stop translation. So apa yang masuk adalah release factor. So release factor is this yellow molecule here. So it is a type of protein. So it binds directly to the stop codon in the A site. Okay, so the function of this release protein is to hydrolyze the bond between the tRNA in the P site and the last amino acid of the completed polypeptide chain. So the polypeptide is released. So the function of this release factor is to break the bond here, the bond between tRNA and the last amino acid of the polypeptide chain. So after the polypeptide is released, so and then the whole association of this structure, okay, will be dissociated, and then will be separated lah. Okay, so the two ribosomal subunit and other components of the translation assembly will dissociate. All of the component will dissociate. Okay, so that is termination. Okay, so this is polyribosomes. So polyribosome is when one strand of mRNA, okay, digunakan dengan digunakan oleh banyak ribosome to make many proteins here. Okay, so in it says here in bacteria and eukaryotes, multiple ribosome translate an mRNA. Nampak? An mRNA. Many ribosome translate one single mRNA at the same time. So that is polyribosomes. So this produce many copies of the same polypeptide chain simultaneously dengan serentak. So dia boleh menghasilkan protein yang sama dengan serentak sebab banyak ribosome baca one single mRNA. Okay, so that is polyribosome. So ingat kat sini 5 prime. Sorry. So this is the 5 prime end. So this is the 3 prime end. Okay, so this diagram is to summarize the whole process of protein synthesis. Okay, so as you can see from this diagram, so you have the DNA molecule. Okay, so this is the DNA molecule. So at one point, only a certain gene of from that DNA molecule will be used to synthesize certain proteins. Okay, so certain genes, okay, needs to be expressed. So so this gene, for example, in this area, okay, in this region here, will be used, okay, to make the mRNA. Okay, so the mRNA is produced through the process of transcription. Okay, so for this is for eukaryotes, okay, eukaryotic cell. So it means that the mRNA produced now, we call it as pre-mRNA. Okay, so pre-mRNA will undergo the process of RNA processing. So RNA processing involves two major process, which is one, the addition of both ends, okay, which is the 5 prime cap and also the poly A tail. Okay, 5 prime cap and also the poly A tail, and removing the non-coding sequence and joining all the coding sequence. Non-coding, we call it as what? Intron kan? Coding is exon. So you join together all the exon and removes the intron. You don't want the non-coding sequence. So the purpose of RNA processing is to make sure you produce a mature mRNA with a continuous coding sequence, which is a continuous exon, and you produce the two ends here, which is to protect the mRNA from being hydrolyzed in the cytoplasm. Okay, so after that, this mature mRNA will find ribosome here. So you have the small subunit, you have the large subunit. So the mRNA will bind to the binding site of mRNA at the small subunit. Okay, and then after that you have the initiator tRNA, okay, that binds to the start codon of mRNA, and then you have the initiator tRNA that brings the the first amino acid, which is methionine. Okay, methionine. So it sits in the P site, and then you have the large ribosomal subunit that binds to the whole structure. Okay, to the whole structure. So ini adalah dia lah kan. So you have the small subunit, you have the large subunit, and then you have the the E site, the P site, and also the A site. So the P site, you have the tRNA that holds the growing polypeptide chain, the vacant A site that allows the next tRNA that brings the next amino acid according to the codon on the mRNA at the A site. Okay, so once the tRNA that brings the specific amino acid to the A site, so the bond between the last amino acid here, okay, and the tRNA will be broken and make new bonds, new peptide bond between this amino acid and this amino acid. Okay, so next apa dia dalam proses tu adalah translocation. Tadi apa? Peptide bond formation. Okay, so this is the elongation stage. You have the codon recognition, peptide bond formation, okay, and then translocation. Okay, translocation. So that is process translation. The whole process of synthesizing the polypeptide chain is translation. So ingat ya, translation ada 3 stage: initiation, elongation, and termination. Elongation ada 3 cycle: codon recognition, peptide bond formation, and also translocation. Okay, so remember that. So daripada awal semester hari tu, saya minta awak buat mind map. So continue that mind map so that it is easier for you to know the flow, okay, of the whole story. For example, for protein synthesis. So this is a video pasal transcription and translation. I think you all can watch. Awak boleh pergi je dekat dalam YouTube and then type protein synthesis, and then it shows you many animation that relates with transcription and translation. Okay. Awak boleh nampak dia punya pergerakan translocation tu. Tak tahu ni. How do I play this? Okay, sekejap. Saya try play the video. So kalau dengar dengan jelas, okay, just continue listening to the video. If not, you tell me.
We need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme Rna Polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for Rna polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the Rna polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the Rna polymerase slides along the template DNA strand. As the complementary bases pair up, the Rna polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the Rna polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the Rna polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
Okay, so itu dia. So you all boleh cari lagi video dekat YouTube yang lain kan. So ada banyak lagi animation that will help you to understand better anything that you are not sure of. Okay, boleh cari je apa-apa dalam YouTube tu untuk awak belajar. Tapi make sure of valid sources lah. Okay, the information is correct according to what you have read in the book. Okay, so apa-apa bahan yang awak cari dalam YouTube ke whatever, make sure dia tally dengan apa yang awak belajar dalam buku. Okay, jangan cakap dalam YouTube tu is 100% correct. Okay, so it must be true based on the facts in the book. Okay, so now let's proceed to the next subtopic. Ada apa-apa soalan nak tanya so far?
Tak ada.
Tak ada eh? Saya ada attachkan attendance, so I give you the attendance 1 hour earlier, so make sure you sign within this before the end of the lecture. Okay, so now let's proceed to the next subtopic. Okay, so the next one here is on gene regulation and expression. So gene regulation and expression. So regulate is what factors that controls a certain gene to be expressed. What is expressed? It means that the information coded in the gene is used to synthesize protein. That is expression. And express digunakan untuk menghasilkan protein. Certain gene is expressed to produce certain proteins. Okay, and in order to produce that protein, there are factors, okay, that allows that gene to be used at certain point during at certain point lah, okay. Bila yang satu require gene to di-expresskan. Okay, so in this case is gene regulation. Yang the one that we are going to look at as an example is the expression of Lactose Permease, okay, in bacterial cell. Okay, so but first of all you have to understand what is gene expression. So it says here information in the DNA, which is the gene, okay, gene that codes for the synthesis of specific protein is transcribed into mRNA. So the mRNA is then translated into a product. Okay, so the product is protein, and this protein can be enzyme, it can be hormones, or it can be other proteins that is needed by the body. Okay, so it can be proteins that function as structural proteins. Kita belajar itu ada collagen kan, we have microfibrils, microtubules. Okay, so all that are protein and produced through the process of transcription and also translation. Sebab tu kat sini cakap proteins that is needed for specific structural and function of cells. Structure, it forms all that fibers, okay, microfibrils, microtubules, microfilaments, collagen. So that are the proteins that involve in the formation of structure within cell. Kalau function, for example, that it involves in metabolic reactions such as enzyme. Okay, so so the protein needs to be activated lah, kan? So the proteins produced will then will have certain function. So the protein needs to be activated. So activation of the protein. So the main requirement of bacterial gene regulation is production of enzyme and other proteins when needed. Okay. So at certain point of time during the bacterial life, okay, so it needs certain gene to be expressed in order to produce enzyme. Okay, enzymes, for example. So allows bacteria respond rapidly to the changing environmental conditions. So this allows organism to be able to survive lah. So in prokaryotes, the basic mechanism for the control of gene expression is operon. Okay, so what is operon? So operon is a gene complex, okay, or a group of related genes with related functions that are clustered together on the bacterial chromosomes. Okay, so advantage allows the gene to function as one transcription unit, can be controlled and switched on and off together, coordinately controlled. So within the component of operon, you have several regions, and this several region has a specific function. So for example, the region within the operon, you have the promoter for the binding site of RNA polymerase. You have the operator, okay, which is the binding site for repressor protein, and then you have the structural genes. Kan? So I assume you have watched the video, okay, for gene regulation in the YouTube. So for structural gene in the like operon, it has 3 structural gene, which is like Z, like Y, and also like A. Okay, so all these 3 genes, okay, will be used, will be expressed as one mRNA transcript. Walaupun dia ada 3 gene yang berbeza, kan, like Z, like Z, like Y, like A, 3 gene. Dan 3 gene tu dia code untuk for different enzyme. Tapi untuk menghasilkan 3 enzim yang berbeza ni, dia hanya perlu menghasilkan bacterial cell tu hanya perlu menghasilkan 1 mRNA transcript, not 3 mRNA transcript for the 3 structural gene. Hanya 1 mRNA transcript sahaja yang akan dihasilkan. Okay, nampak ni? One transcription units. So allows the genes to function as one transcription unit. So they are clustered together. They are they are side by side. Okay, they are near to one another. Okay, so this is the basic concept, okay, of operon. So you have actually for operon, like operon, it starts with a promoter. Okay, bukan regulatory gene. Regulatory gene is outside of the operon. Cuma regulatory gene ni diperlukan untuk menentukan sama ada structural gene tu akan diekspreskan ke tak. Okay, that is regulatory gene. Okay, so the function of regulatory gene is to basically the information is used to synthesize a protein, which is we call it as repressor protein. Okay, so before it can produce the repressor protein, it has to produce the mRNA lah. Okay, so it copies the information from the gene, regulatory gene, and mRNA is used to synthesize the repressor protein. So this repressor protein will bind to the operator. Okay, so it binds to the operator, and if the repressor protein bind to the operator, so this will not allow RNA polymerase binds to the promoter. So what is the function of RNA polymerase? The function of RNA polymerase is for transcription. is to produce mRNA for the 3 structural genes. So bila dia dah kena block, operator ni block, tak bolehlah nak menghasilkan mRNA, tak bolehlah nak menghasilkan the 3 enzymes from that 3 structural genes. So in this condition, we call it as the operon is switch off. Okay, it is switch off. No mRNA will be produced, no protein will be produced. So this only occurs at specific condition. So in case of like operon, dia akan berlaku in the absence of lactose. Okay, when lactose is absent, then the operon is switched off because repressor protein binds to the operator and blocked. Okay, block and prevent RNA polymerase from binding to the promoter. That is in the absence of lactose. Okay. So it says here operon is the entire stretch of DNA required for enzyme production. So operon ni dia start daripada promoter promoter, operator and the 3 structural gene in the example of like operon. So kita ada banyak, dia ada banyak operon sebenarnya yang kita nak tengok dalam operon syllabus hanya untuk like operon. Okay, like stands for lactose. Okay, lactose. The how the bacterial cells use lactose. Okay, use the lactose to undergo process of cellular metabolism. Okay, lactose, as you know, it is a disaccharide. Okay, so it needs to be broken down into its monomer, which is glucose and galactose. So once you have produced the monomers, then the monomers, which is the glucose, for example, can be used during cellular respiration, okay, to produce energy, okay, in the form of ATP. Okay, so operon. Operon consists of promoter, operator and structural gene. Regulatory gene is not included. Okay, remember that. So dia dekat depan promoter. It is outside of the operon. So promoter is the binding site for RNA polymerase to initiate transcription of the structural genes. Okay, so promoter is the binding site of RNA polymerase used to synthesize mRNA later. Okay, so operator is the on-off switch in the segment of DNA that controls the synthesis of mRNA, on-off. Okay, so kalau ada repressor, so the operon is switched off. Kalau tak ada repressor, so the operon is switched on. Okay, so bila masa tak ada repressor ni, it is when lactose is present. Okay, and lactose, some of the lactose will get converted into its isomer, which is allolactose. So this allolactose binds to the repressor protein and causes the repressor protein to change shape. So bila dia bertukar bentuk, so maksudnya dia tak boleh lah nak bind kepada operator. So bila dia tak boleh nak bind kepada operator, apa jadi? So the operator is free. Okay, so now when the operator is free, so the operon is switched on because the RNA polymerase can now bind to the promoter and make or synthesize the mRNA for the 3 structural genes here. Okay, so that is in the absence of lactose. Lactose causes operon to switch off. Okay, sorry, switch on. Causes the operon to switch on in the presence of lactose. So regulatory gene is located outside. Nampak highlight kat sini. Regulatory gene is located outside and upstream of the operon. Regulatory gene codes for the synthesis of mRNA that is translated into repressor protein. Repressor protein binds to the operator of a particular operon. So there are many operon. So in this case, we are only going to look at like operon. Okay, so like operon, so it stands for lactose operon. So lactose dalam bentuk italic lah, and so because we have many different operons. So in E. coli, which is a bacteria, lactose operon codes for the enzyme that function in the uptake and metabolism of lactose. So the three structural genes that is transcribed into a single mRNA. You have three genes, and the three genes will be transcribed into one mRNA strand, okay, which is then translated into three different enzymes. Okay, so like one, which is the regulatory gene. The name of the regulatory gene is like one here. Okay, for like operon codes for the synthesis of mRNA, which translated into repressor protein. Okay, like repressor. So allolactose, which is an inducer to the repressor protein of like operon, is an isomer of lactose formed in small amounts from lactose that enters into the cell. So kalau baca dalam buku, for example, ni adalah dia ada satu eksperimen that was carried out, and in the experiment it involves, okay, the E. coli, okay, being cultured. It is being cultured. Imagine this is petri dish, contohnya, okay. So the medium here, the medium in the petri dish, it has lactose, okay. Selalunya bakteria ni tak guna dalam keadaan yang normal. The E. coli does not use lactose for its normal metabolism, okay. But in this case, you induce the expression of this like operon gene, okay, the genes in the like operon. So you grow the bacteria in a medium that contains lactose. Okay, awak tak letak apa-apa yang lain. So lactose ni adalah sumber makanan untuk bakteria ni. So in order for the E. coli to use up the lactose, the like operon, the gene in the like operon will be used. Okay, so it means that the operon will be switched on in the presence of lactose. So lactose ni dia akan masuk dalam bacterial cell ni, dan sebahagian, okay, it says here a small amount of the lactose that enter into the cell will be converted into its isomer, which is allolactose. Allolactose function as an inducer. Okay, inducer, dia induced. Kan sama ada transcription boleh berlaku ke tak? Operon ni switch on ke tak? Faham? Okay, inducer, dia bertindak sebagai inducer. Dia menentukan operon ni switch on or not. The genes, the three genes will be expressed or not. Okay. So the structural genes here is the like Z. Ingat ya, turutan dia mesti start dengan like Z. After the operator, you have the like Z gene, you have the like Y, and then like A. So function of like Z gene is to produce the enzyme beta galactosidase. Okay, so the function of beta galactosidase is to hydrolyze lactose into glucose and galactose, okay, which are utilized for cellular metabolism. So once you have break down lactose is a disaccharides into its monomer, glucose and galactose, then only the bacterial cell can undergo the normal cellular metabolism, cellular respiration to produce energy in the form of ATP lah. So this allows the bacteria to live on, okay, because it has energy to live on. Okay, so and then like Y is lactose permease. What is the function of lactose permease? Is to facilitate the uptake of lactose into the cell. So macam mana lactose ni nak masuk dalam sel? Mesti ada enzim yang membolehkan dia masuk dalam sel. So in this case adalah permease, permit, lactose permease. Lactose boleh masuk ke dalam sel dengan terhasilnya enzim lactose permease yang dihasilkan oleh enzim, so yang dikod oleh gene ni, like Y, okay, like Y. And then like A, produce the enzyme transacetylase. So the function of this enzyme is to detoxify other molecules entering the cell via permease. So maksudnya dengan terhasilnya transacetylase, hanya lactose sahaja yang boleh masuk ke dalam cells, okay, melalui permease, enzyme permease tu. Okay, permease. So permease are located at the, once it is produced, it is then embedded in the membrane of the bacteria. Okay. Okay, so next, okay, so in the presence of lactose, so sekarang ni kita nak tengok keadaan presence of lactose. So ini adalah keadaan yang pertama. Lactose is present. Okay, is present. So allolactose acts as an inducer and binds to the repressor proteins. So this causes the repressor protein to change shape into inactive form. Okay, so nampak keadaan dia sebelum dan selepas dia bind kepada allolactose. Berbeza kan? So bila dia dah berubah bentuk, so the repressor protein cannot bind to the promoter. So now the operator is activated and the like operon is switched on. Okay, so lactose present, like operon switch on because repressor protein changes shape due to its binding to the allolactose. So operator is now free. Okay, so RNA polymerase can bind to the promoter. So transcription of the three structural genes, okay, structural genes are like Z, like Y, and like A, produces a single long mRNA. Walaupun ada 3 gene yang berbeza here, dia hanya akan menghasilkan satu sahaja mRNA. Only one single mRNA transcript will be produced through the process of transcription. So after you have produced a single mRNA here, and so setiap gene ni dia akan ada start and stop codon dia lah. So itu yang menentukan dia boleh hasilkan 3 enzim yang berbeza, bukan satu molekul yang menjadi 3 enzim kan. So it produces a separate enzyme due to the presence of start and stop codon. Okay, setiap gene ni dia ada start and stop codon, start and stop codon, start and stop codon. So on the mRNA it has this region, start and stop codon, to produce and synthesize the 3 separate enzymes. So translation of the mRNA produces 3 separate proteins, okay, for metabolism of lactose, namely beta-galactosidase from like Z, lactose permease from like Y, and transacetylase from like A. You have, ini adalah antara soalan favorite kalau nak keluar lah dalam exam, okay. So dia akan tanya what are the genes, okay, what are the 3 genes, what are the enzymes produced by the 3 genes, and what are the functions of the 3 enzyme here. So you have to describe carefully lah. Dulu kalau macam salah careless lah cara jawab adalah untuk yang function permease ni. Okay, mesti ada perkataan facilitate the uptake of lactose into bacterial cell. Okay, bukan macam mana tak itu jawab, tapi salah lah, mesti ada perkataan like facilitate the uptake. Okay, facilitate the uptake of lactose into bacterial cell. You have to remember for permease. Okay, ini yang selalu salah, function permease ni. The beta-galactosidase is easy, okay, to hydrolyze lactose into glucose and galactose. Okay, transacetylase jarang ditanya, tapi ingat jugalah. Okay. Okay, so the next situation is the absence of lactose. Okay, the next situation here is lactose is absence. There is no lactose. So in the absence of lactose, obviously there's no allolactose. When there's no allolactose, there will be no molecule to bind to the repressor protein lah. So repressor protein can happily bind to the operator. Okay, so this causes the repressor protein bind to the operator and forms the repressor operator complex. Okay, the repressor operator complex over here. So this blocks part of the promoter. So this prevent the RNA polymerase binding to the promoter. Okay, so ada buku yang saya baca, actually RNA polymerase ni dia memang bind kepada promoter, tapi dia akan extend sikit pada bahagian operator sini. Dia bukan exactly bind kepada promoter region je, dia akan bind sikit ke dalam operator region. So that's why kalau ada repressor protein bind kepada operator, memang tak boleh langsung RNA polymerase bind kepada promoter. Okay, so like operon is switched off, structural gene is not transcribed, no enzyme will be produced. Okay, the 3 enzyme cannot be synthesized. Okay, so like operon. So in the context of gene regulation, like operon is a negative control of genes, okay, because the operons are switched off by the active form of repressor protein. Okay, negative control. Maksudnya kalau ada repressor protein bind kepada operator, then the like operon is switched off. So like operon is an inducible operon. It needs an inducer. Inducer in this case adalah allolactose, which is isomer of lactose. So like operon is an inducible operon, an operon that is always off but can be induced, okay, to be on when a small and specific molecule interacts with different regulatory protein. Okay, regulatory protein in this case is the repressor protein lah. Okay, so enzyme of lactose pathway, which is inducible enzymes. Enzyme of lactose pathway is an inducible enzyme, okay, because their synthesis is induced by the presence of allolactose, okay, a chemical signal which is allolactose. So enzyme usually function in catabolic pathway, break down a nutrient into a simpler molecules, okay. Lactose, a disaccharides, perlu di breakdownkan kepada monomer dia, which is glucose and galactose. So enzim-enzim yang terhasil pada like operon ni digunakan untuk catabolic pathway. You break larger molecule into smaller molecules. Okay. So advantage, cells avoid wasting resources by producing enzymes when only the nutrient is available. So dalam keadaan normal, E. coli tak menggunakan lactose. Tapi bila awak culture dia contohnya dalam medium yang ada lactose, then only this like operon will be switched on. Okay, it is an inducible operon. Dia hanya akan di-on-kan bila certain situation berlaku. In this case adalah dengan adanya inducer which is allolactose. Okay. So kita try play video ni berapa minit dah? 5-6 minit boleh lah kot. Okay, sekejap. Kalau tak dengar video, bagi tahu just bagi tahu dengan mic awak.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous, like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point may not be transcribed at all.
I need to disconnect. Sampai awal Veron ya. Saya ribet dulu ya. So I'm going to play that start of yang dia explain pasal Operon lah.
Here. First, RNA polymerase. It's called an operon eukaryotes. A few eukaryote exceptions. Build up the substances that they act on. Now let's talk. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is order transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor. So let's take a look at our setup here. This is an example called a Lac operon. Notice there's a promoter region of the DNA, the operator region of DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? it has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so far ada soalan? Any questions? So kalau tak ada soalan, saya harap semua faham lah. Okay, so now we are going to proceed with the next subtopic, which is on mutation. Okay. So mutation, so this is the definition for mutation. It is a change in the nucleotide sequence of an organism's DNA. Okay, so in your DNA, you have many nucleotide sequence. Okay, so when the process of, for example, transcription that produces new genetic materials for the new cells, akan ada tendensi untuk berlaku mutation. Dia akan berlaku ada tendensi untuk berlaku mutation that change the nucleotide sequence of the DNA, or it can change mutation can also change the number of chromosome in the cell. It can change the structure of the chromosome. Okay, so these are all the different types of mutation that can occur to an organism's genetic materials. Okay. So mutation provides the allelic variations that are responsible for the huge diversity of genes found in among organisms. Okay, so contohnya kalau genes that code for your skin pigmentation, kan? Skin pigmentation adalah disebabkan oleh boleh terhasilnya pigment melanin. Okay, so your skin have melanin. Okay, so bila berlaku mutation pada gene untuk yang menghasilkan melanin, enzim yang sepatutnya menghasilkan pigment melanin tak boleh nak dihasilkan, terhasillah yang recessive allele dan boleh menyebabkan penyakit albino, contohnya albino tu. Okay, so that is allelic variation. Okay, so normal gene kan boleh menghasilkan melanin. Okay, yang mutated recessive allele cannot produce melanin. So kalau orang tu dalam keadaan recessive, homozygous recessive, so dia dalam keadaan berpenyakit, dia ada albino lah, faham? Okay, cannot produce pigment melanin. Okay, so this is the mutation boleh menghasilkan different forms of allele for certain genes, kan? So ada recessive allele, ada dominant allele, Itu adalah salah satu faktornya disebabkan oleh berlakunya mutation, produces many variations of alternative form of a gene, okay, alleles. It produces different alleles for a gene. So if mutation occurs in the reproductive cells, it can be passed from parents to offspring and the future generations. Okay. So kalau macam contoh yang paling senang, nak tengok mutation ni dulu saya buat masa master adalah you radiate seeds, okay, plant seeds, you radiate the plant seeds and then you grow the seeds to produce the offspring, and then you observe lah whether the seed produces a new phenotype that is different from the parents. Okay, so you grow the seed for many generation to make sure that the new phenotype maintains. Itu salah satu cara cara untuk menghasilkan new alleles to produce new phenotype. Excuse me. Okay, so change in the structure of DNA leads to changes in the sequence of bases in the mRNA, which leads to the change in amino acid sequence of the polypeptide change and change in the function of the protein. So bila awak tukar sequence pada pada DNA, obviously lah sequence pada mRNA pun akan berbeza. So when you have a different sequence on the mRNA, obviously you will produce a different sequence of amino acids of the proteins. Okay, so when the protein sequence of amino acids, the protein that's supposed to be, for example, enzyme, so the enzyme is a different function within the organism. It may produce a different phenotype for that organism. Okay, so mutation boleh jadi negatif, okay, ataupun dia tak ada efek pun. Tak besar dia berlaku organism tu. So dia boleh ada tiga efek lah, negative, neutral or beneficial. Okay. So large scale mutation and small scale mutation. So large scale mutations include chromosome mutation. Okay, so chromosome mutation ni maksudnya awak tukar the number of chromosome within the genome of that organism can change. Contohnya Down syndrome. So instead of each cell have 46 chromosome, they have 47 due to an extra chromosome at chromosome 21 contohnya. Alright, or changes to a large section of chromosome. So a section of the chromosome may be deleted. So when a section of chromosome is deleted, that section obviously it contains many genes. Okay, it contains many genes, and when the gene is removed or deleted, obviously the gene cannot be expressed for the function of that cells. Okay, so deletion can have a detrimental effects towards that organism. Okay, so that is for example large scale mutation. For small scale mutations, okay, it involves changes in the nucleotide pair sequence. Okay, so it can involve changes in one nucleotide pair or a few nucleotide pairs. Okay, so this include point mutation. So point mutation, it only involve changes in a single nucleotide pair of a gene. Satu je pairing dia bertukar. Okay. Pair tu kenapa? Sebab DNA double strand kan, so mesti ada pairing antara the first strand and also the second strand. So that's why kita panggil dia sebagai base pair, okay, pairing. So two categories of point mutation within a gene is single nucleotide pair substitution, tukar kan, daripada contohnya sepatutnya A, tukar dia jadi G. Okay, so this produce a different sequence of DNA. So or nucleotide pair insertion or deletion. nucleotide pair insertion and deletion effect dia lagi teruk daripada nucleotide pair substitution. Sebab apa? Once you delete even one single pair, okay, so dia akan menyebabkan apa? Frame shift mutation. Semua amino acid untuk protein tu, the point bila berlakunya deletion tu akan berubah. Sebab dia menyebabkan frame shift mutation to occur. When a frame shift mutation occurs, it produces or it codes for a different amino acid all the way through the production of the polypeptide chain. Okay. Tapi kalau single nucleotide pair substitution, it may code for a different amino acid, or it may it may change the codon but still code for the same amino acids. Okay. Ataupun dia boleh tukar boleh code for amino acid tapi dia tukar pada stop codon. Bila dia tukar pada stop codon tu yang akan teruk. Jadi teruk lah. Sebab apa? Sepatutnya dia hasilkan polypeptide yang lebih panjang. Tapi bila jumpa dia bertukar jadi stop codon, so it terminates the process of protein production tu. Okay, it terminates the process of translation. Okay. So next, okay. So types of small scale mutation. You have the nucleotide pair substitution and nucleotide pair insertion or deletion. For nucleotide pair substitution, it is the replacement of one nucleotide and its partner with another pair of nucleotides. Okay, so this leads to three types of mutation, which is silent mutation, missense mutation, and also mutation. So for nucleotide pair insertion or deletion, so are addition or losses of nucleotide pairs in a gene that may alter the reading frame, okay, of the genetic message codon and also the codon on the mRNA during translation. So this leads to frame shift mutation. Or masukkan satu extra nucleotide ataupun delete satu extra nucleotide, dia akan menyebabkan frame shift mutation. It codes for a totally different amino acid throughout the production of the proteins. Okay, so kita tengok yang example yang Okay, sorry saya terdisconnect. Still on silent mutation ya tadi. Saya stop sampai mana saya explain tadi? Kelas?
Frame shift mutation.
Silent saya tak masuk. Maksudnya ada saya terdisconnect bila saya start explain silent mutation.
Yeah, madam.
Alright. Okay, so kita tengok balik semula. We are going to look at again on silent mutation. Okay, so basically for silent mutation, as you can see, it occurs, for example here. Okay, so this is the DNA template of the wild type. Wild type, it means that it is the original DNA. Okay, original DNA without any mutation occurs to that DNA. Okay, wild type. Organism yang asal tanpa berlaku mutation dan menyebabkan penghasilan new phenotype. Okay, dia yang asal. So that is wild type. So this is the DNA template strand. So for example, this DNA template strand, the top strand here will become the template to synthesize the mRNA. Okay, that is used to synthesize this polypeptide chain that consists of the amino acid methionine, lysine, phenylalanine, and also lysine. So at certain point, okay, mutation occurs that involve nucleotide substitution. Okay, so kalau untuk silent mutation berlaku, tengok kat DNA template ni, instead of dia baca CCG, it changes guanine into adenine. Okay, so this is a codon, the three nucleotide pair. Okay, so instead of A, it changes to A, and obviously
Madam Syakirin. Madam Syakirin? Tak ada.
okay, once again saya terkeluar. So ini maksudnya petanda-petanda nak kena stop kot. Tapi saya nak habiskan juga pasal mutation. Dah ada lagi about 15 minutes. Sorry. Okay. Ini sebab saya guna wireless lah ni. Saya guna wifi. Alright. Okay, so for silent mutation basically dia berlaku disebabkan oleh perubahan yang berlaku pada base yang ketiga of the codon. So wild type base yang ketiga berubah kan, so 1, 2, 3 base yang ketiga berubah. Instead of guanine dia code for adenine. Okay, so obviously when you change the DNA template you will change the sequence of the mRNA. So tapi dia bernasib baik, you are fortunate because when you change the third base, instead of reading GGC, it reads GGU. Okay, because the change only occurs at the third base, this still codes for the same amino acids. Okay, perubahan berlaku pada base yang ketiga. Ini berlaku disebabkan oleh apa? Sebab you recall back on feature of genetic codes. Kan, feature of genetic codes is one of it adalah redundancy. What is redundancy? Redundancy, if you refer to the codon table, mRNA codon table, some of the mRNA are coded by 4 codons, okay? Some mRNA are coded by 2 codons, some are coded by 1 codon. So dia beneficial pada amino acid yang coded by 4 codon, for example, okay? 4 codons, 4 codons. Contohnya, in the case of, apa nama? Okay, glycine kan. So kod yang asal dia read GGC. Tapi bila berlaku perubahan pada base yang ketiga, instead of GGC, dia read GGU. Okay, still code for the same amino acid sebab it is due to the redundancy feature of genetic code. So this redundancy will prevent missense mutation to occur. Missense mutation is when you change the codon of mRNA, it codes for a different amino acid. Tapi bila adanya redundancy, dia boleh menyebabkan silent mutation berlaku yang menyebabkan still the codon still code for the same amino acid. Okay, disebabkan perubahan pada base yang ketiga. Okay, this is the redundancy of genetic code. So for the next one is missense mutation. So you change the codon. Sorry, there is a mutation that occurs, okay, in the DNA template. Instead of reading CCG, it changes to TCG. Okay, you change the first base. Okay, so of the codon. So when you change the DNA, obviously you change the DNA the mRNA sequence kan, instead of baca GGC dia baca AGC. Okay, instead of coding for glycine, it changes to a different amino acid which is serine. Okay, so this is missense mutation. So ada missense mutation walaupun dia ubahkan satu je amino acid, dia boleh menyebabkan penyakit. Contohnya apa? Penyakit apa tu? Yang anemia. When you changes the structure of the hemoglobin, okay, you change the structure of the hemoglobin because you change one amino acid to another amino acid. So this cause the hemoglobin will not be able or capable of binding to oxygen. So your red blood cells cannot carry enough oxygen. Okay. Apa penyakit dia? If you all ingat, kita tengok dekat extension of Mendelian genetics hari tu.
Sickle cell trait.
Sickle cell, kan? Sickle cell. So sickle cell tu disebabkan oleh mutation ni lah, missense mutation. One amino acid is changed to another amino acid yang menyebabkan perubahan pada structure hemoglobin tu. Okay, so bila hemoglobin berubah structure dia, it cannot bind to enough oxygen. Red blood cells cannot bind to enough, cannot carry enough oxygen. Okay, so itu menyebabkan sickle cell disease. Alright. Tapi ada juga bila mutation ni tak menyebabkan perubahan yang sangat ketara lah, bila amino acid yang diubahkan tu mempunyai ciri yang sama dengan ataupun sifat the same chemical, okay, the same chemical property as the amino acid, as the original amino acids, okay. So itu kalau tak menyebabkan perubahan yang sangat ketara, bila amino acid yang baru mempunyai similar chemical property as the original amino acids, okay, ataupun kedudukan amino acid bukan dalam kedudukan yang penting, macam contohnya dekat active site kan. So bila perubahan tu tak berlaku pada active site of enzyme, dia dekat tengah-tengah molekul enzyme tu, so dia tak menyebabkan perubahan yang ketara pada function protein tu. So ada yang memberi kesan yang teruk, ada yang tak memberi kesan yang obvious, yang teruk. It depends, okay. It depends on the function and the structure of the proteins, okay. Okay, so the next one is nonsense mutation. So for nonsense mutation, sepatutnya the code for the codon, okay. So the codon of the mRNA codes for amino acids. AAG in this case is lysine. So if mutation occurs, instead of it reads AG, it reads AG. So terus stop the process of translation. So that is nonsense mutation. Changes an amino acid codon to a stop codon. So this will obviously terminate the process of translation and no protein can be produced, or the protein produced is not functional. Okay, it cannot produce a functional protein. Alright. So resulting in a shorter polypeptide. Okay, so the next one is frame shift mutation. So frame shift mutation is due to either one, deletion or insertion. You insert an extra nucleotide, or you delete a nucleotide pair. So this will alter the reading frame of the genetic message on the mRNA. So when you alter the reading frame, obviously all the way through the point yang mana berlaku mutation, in this case as you can see, you delete A kan. Asalnya dia read A and code for the amino acid phenylalanine. Okay, the next one is CCG. It codes for the glycine, for example. But if you delete, okay, if you delete A, so obviously it reads instead of AAA, dia akan read AAC. Dia bawa ke depan C ni, kan? So that is frame shift mutation. It alters the reading frame. So dia akan baca instead of UUU, dia jadi UUG. G of glycine dia bawa ke depan. So dia akan obviously alters all the way through the reading frame of the mRNA, produces a different sequence of amino acids, okay, for that protein. So kesan dia lagi teruk lah daripada yang the previous mutation that we have looked earlier, okay. Sama juga if you insert a nucleotide pair, kan, instead of TTC dia baca ATT. C ni dia bawa, dia anjakkan kedudukan dia kepada next codon. Okay, so this will alter the reading frame of the mRNA, produces a different sequence of amino acids. Okay, tapi berlainan cerita kalau terus insert, you insert 3 nucleotide pair, or you delete 3 nucleotide pair. So it means you insert a new codon or you delete a new codon. So dia hanya akan affect one amino acids, kan? Sebab one amino acid is coded by 3 nucleotides ataupun a codon. Okay. So delete, for example, the sequence TTC, so the codon AAG will be deleted. Okay, so instead of ada lysine antara methionine and phenylalanine, you delete AAG, so means that there is a missing lysine in between. So kesan dia tak seteruk macam frame shift tadi lah. Walaupun tadi you delete only one nucleotide pair, or you insert one nucleotide pair, it causes frame shift mutation. Tapi kalau yang ini or delete or insert 3 nucleotide pair, it just affects one amino acid. Whether a new amino acid to be added or one amino acid to be removed. okay, from the amino acid sequence. Okay, so this is the whole mutation lah, jenis mutation yang kita dah tengok tadi. Okay, so for the next one is chromosome mutations. Okay, so large-scale chromosomal alteration can affect an organism's phenotype. So error in meiosis or damaging agents such as radiation can cause breakage of chromosome, which leads to 4 types of changes in chromosome structure, which is deletion, duplication, inversion, and translocation. Saya nak tanya, lepas ni korang ada kelas ke? S9 and S10?
S9 tak ada.
S10?
Tak ada.
Saya nak habiskan skin je. Nak habiskan mutation je. Alright? Okay. Okay, thank you. Okay, so for types of mutation, okay, which 4 types of changes in chromosome structure adalah deletion, okay, duplication, inversion, and also translocation. So kita tengok dulu yang untuk deletion. Okay, so deletion is the deficiency in a chromosome resulting from the loss of a fragment through breakage. Okay, so the affected region is then be missing certain genes. So for example, you delete this segment D. Obviously D ni dekat region dia will have several other different genes that code for other proteins, that code for specific proteins kan, that involved in the expression of certain phenotype. Okay, so let's say lah kat sini dia ada gene untuk code for the production of skin pigmentation. So nanti tak adalah gene tu, faham tak? Okay, so that is deletion. Kalau dia code, kalau kat sini code for the production of your legs and arms. So kalau tak ada region ni, tak adalah awak hasil kaki dengan tangan kat situ, faham? Okay, so that is the effects of deletion in the chromosome segment. Same goes to the duplication. So duplication of a portion of a chromosome resulting from fusion with a fragment of a homologous chromosome. Okay, so dia macam ni kalau nak faham. Dia selalu kalau duplication and deletion ni dia berlaku secara selari. Dia sebab dia melibatkan homologous chromosome. So let's say lah this is a homologous chromosome. And homologous chromosome that you inherit from your father and also from your mother and also your father kan. So you are a diploid organism. Diploid but you have 2 chromosomes, a pair of chromosomes, okay, for each chromosome that you have. So homologous chromosome obviously they carry the same genes, can carries the same genes, alleles of the same gene that you inherit from your parents. Okay, so for example, if duplication occurs and duplication and deletion occurs, okay, let's say lah chromosome yang pertama ni, this segment is deleted. Okay, segmen ni dia pergi mana? Dia boleh ditukar kepada dia punya homologous pair dia. So dia punya produk dia akan jadi satu deleted, satu pair delete, satu pair mengalami duplication. So satu pendek kan jadi pendek sebab missing a segment. Satu jadi lebih panjang. Faham? Kan sebab asalnya lebih panjang. Asalnya kat sini so dia akan pindah ke sini. Okay. So one is deleted, one will be duplicated. Okay. So this chromosome is it experience deletion. This one it experience duplication. Okay. Okay, so the next one is inversion. Okay, so still under alteration of chromosome structure. So inversion resulting from reattachment of a chromosomal fragment in a reverse orientation to the original chromosome. So original chromosome segment dia baca A, B, C, D kan. So these different regions have different genes. Okay. Tapi bila berlaku inversion, awak ubah kedudukan segmen tu. Kan, you invert the segment. So this will gene tu still ada pada kromosom tu, tapi kedudukan dia dah berubah, inverted. Okay. So this will also affect the expression of the gene, sebab the expression of genes is also influenced by its location. Okay, so walaupun gene tu tak hilang daripada kromosom tu, tapi dia akan juga mengganggu ataupun mempengaruhi cara macam mana gene tu diekspreskan. Faham So that is inversion. And then for translocation, a translocation moves a segment from one chromosome to another non-homologous chromosome. Tadi kita cerita pasal homologous chromosome kan, dia mengalami deletion and duplication. Tapi kalau for translocation, it involve non-homologous chromosome. Contoh yang kita akan tengok nanti, okay, kromosom 9 dengan kromosom 22. They change segment. Okay, so they are non-homologous chromosome. Non-homologous chromosomes are different chromosomes that carry different genes. Okay. Then we have how many pairs of chromosome? 23 kan. So those 23 pairs are non-homologous chromosome. They carry different genes. Okay. So as you can see from this example, you have two true chromosomes. Contohnya ini adalah kromosom 1, ini kromosom 2. Okay. So segment of chromosome 1 and chromosome 2, they change segments. Okay, they change segment. Walaupun gene tu still ada dalam genome organism, tapi disebabkan oleh perubahan kedudukan gene tu, dia juga akan mempengaruhi cara macam mana gene tu akan diekspreskan. Okay. So translocation moves a segment from one chromosome to a non-homologous chromosome. Okay, so human disorder due to chromosomal alteration. Okay, so alteration of chromosome number and structure are associated with a number of serious human disorder. So you have the Down syndrome, Klinefelter syndrome, and also Turner syndrome. So the first three examples of human genetic disorder is due to the alteration of chromosome number. Kan, Down syndrome, extra chromosome at chromosome 1. Klinefelter dia dekat sex chromosome, and also for Turner syndrome dia berkaitan dengan sex chromosome XY tu. For the next example, for the next human disorder adalah cry-du-chat and also chronic myelogenous leukemia, which is a type of cancer. So cry-du-chat disebabkan oleh deletion at chromosome 5 kan. So chronic myelogenous leukemia disebabkan oleh translocation, okay, antara kromosom 9 dengan 22, okay, kromosom 9 and 22. Okay, so for Down syndrome or trisomy 21, so we have in our genome, okay, we have 23 pairs of chromosome. Okay, we have 23 pairs of chromosome. Yang ke-23 adalah your sex chromosome lah. Kalau lelaki XY lah, kalau perempuan XX. But at chromosome 21, you have an extra chromosome. Instead of only 2, you have 3. Okay, so Down syndrome ni kalau belajar detail genetics oleh berlakunya mutation tu masa proses meiosis. Masa proses meiosis, semasa menghasilkan eggs pada perempuan. Dia melibatkan homologous chromosome cannot separate. Kan, homologous chromosome kita belajar, homologous chromosome masa proses anaphase akan separate into two new daughter cells. Tapi disebabkan oleh the homologous chromosome cannot separate, telur tu instead of chromosome 21, dia ada satu je chromosome, dia ada dua. Faham tak? Telur, ovum, and your ovum. So kromosom 21 ni sepatutnya masa proses anaphase dia separate kan. So dia akan separate, telur tu akan receive satu je kromosom 21. Okay, tapi disebabkan oleh berlakunya mutation, telur tu instead of dia jadi macam ni, dia akan homologous chromosome 21 ni cannot separate. So that's the egg. Kan? So bila egg ni dia difertiliskan oleh sperm yang bawa satu je kromosom 21. So bila berlaku fertilization, jadilah individual yang mana bila kromosom 21 dia pair, dia ada 3 kromosom 21. Dia ada 3. Okay? So that is trisomy 21. Sebabkan oleh berlakunya mutation pada proses meiosis pada perempuan. Okay? Okay, so it is result from an extra chromosome 21. So each body cells has a total of 47 chromosome. So the cells are trisomic for chromosome 21. Okay. So features of Down syndrome individuals have characteristics, facial features, lantern eyes, short neck, short stature, small heads and eyes, long tongue, heart defects, susceptibility to developmental delays. So prone to developing leukemia and also Alzheimer. On average, have a life span shorter than normal. Most are sexually underdeveloped and sterile. Okay. Ini soalan ni pernah keluar dalam exam yang features of Down syndrome ni. So, take note lah. Pernah keluar. Tak maksudnya dia akan keluar lagi, tapi pernah keluar. So, itu adalah antara potensi soalan-soalan yang boleh ditemukan lah. Okay, so Klinefelter syndrome, a type of disorder where a male has XY, tapi kalau Klinefelter syndrome dia XXY. Okay, so occurs once in every 2,000 live birth. Okay, so have male sex organ but the testes are smaller. Okay, so because the testes is not developed well, so they are sterile. Okay, sterile tu masih mandul lah. And then the X chromosome is inactivated but has some breast enlargement and other female body characteristics. So for Turner syndrome pula, monosomy X. Okay, so Turner syndrome. Dia perempuan. Okay, the person is a female, but that person, the female person is missing. Dia instead of XX jadi XO. Okay, jadi XO. So they are XO individual with female phenotypes. So they are sterile because of their sex organ do not mature. Some are normal. Okay, have normal intelligence. So but when you provide estrogen replacement therapy, so they develop female secondary sex characteristics. So kena tolong sikit lah dia give the hormone estrogen. Okay, develop the female secondary sex characteristics. So the last two disease is Chromosome. So Chromosome dia involve di chromosome 5. Okay, so addition, so this is chromosome 5, so dia mengalami deletion. Okay, so the child born with this deletion is severely intellectually disabled, has a small head with unusual facial feature, has a gait that sounds like a meowing of a distressed cat. So die in infancy or early childhood. So selalunya kalau anak yang ada keadaan ni dia tak boleh nak survive. Okay, dari survive to adulthood. So selalunya akan meninggal pada waktu kecil lah. Okay. Awak boleh cari dekat dalam internet ke dalam YouTube. Macam mana cry the chat ni, macam mana bunyi meowing pada kimia. Okay. So this in the cell that will become white blood cells. Okay, dia akan berlaku pada sel yang akan jadi white blood cells. Okay, so this is due to the reciprocal translocation. Reciprocal tu maksudnya the saling exchange segment. Okay, saling exchange segment involving the exchange of large portion of chromosome 22. Okay, chromosome 22, large segment of it is changed with the small segment of chromosome 9. So this produces a So in this case dia menyebabkan boleh berlakunya kanser. Okay. Okay, so we end our lecture for today. So do you have any questions?
Madam,
Plan?
boleh saya
Ya.
tanya